When researchers drilled into Alaska's permafrost, they expected bones, maybe preserved fur. At first, that's exactly what they found. But then, something didn't add up. Tissue still holding liquid blood after 40,000 years. A virus frozen for 30,000 years that woke up in a lab and began killing cells. No one can fully explain what's been buried under Alaska's ice. And the deeper they look, the stranger it gets. Cold is a ruthless editor. It strips away the unnecessary, leaving only what can survive. Under Alaska's permafrost, time does not just slow, it nearly stops. At first, what is preserved seems straightforward: bone, fur, the hard parts that resist decay. But then, deeper in the ice, something unexpected appears. Muscle fibers still red. Collagen strands that look as if they were woven yesterday. Sometimes, even pockets of fluid, thick and dark, linger where blood once flowed. This is not ordinary freezing. At temperatures below -5 degrees Celsius, the rules of biology change. Water inside cells can slip past the familiar ice crystal stage and transform into a glassy solid, a process scientists call vitrification. No shards to rupture membranes. No slow grind of decay. Just a perfect, silent pause. Minerals in the soil add another layer of protection. Silts and clays buffer the pH, locking out oxygen that would normally fuel rot. The ground itself becomes a vault, holding tissues in a kind of chemical stasis. Even after 40,000 years, collagen, the protein that gives skin and tendons their strength, can remain intact, its triple helix still tightly coiled. Some animals, like the ancient foal found in Siberia, show blood that resists freezing even at -17 degrees Celsius. Scientists suspect natural antifreeze proteins, or perhaps a cocktail of sugars and salts, lower the freezing point just enough to keep fluids from turning solid. In this deep cold, enzymes that would break down flesh simply stop working. Bacteria and fungi cannot thrive. Everything slows, then halts, as if the clock itself has been buried in ice. This is the chemistry that makes the impossible possible. It is why tissues, and sometimes even liquid blood, can survive for ages beneath Alaska's frozen ground. But the ice does not just preserve. Sometimes it hides things we are not ready to find. A stainless steel corer bites into the ice. Every surface has been sterilized, every move tracked. The sample, 30 meters down, looks unremarkable. Just ancient soil, frozen since mammoths roamed above. But inside the clean room, under filtered air and ultraviolet lights, the routine breaks. A technician slices away the outer layer, exposing a core that is darker and denser than expected. The protocols are strict. Gloves changed twice. Tools passed through ethanol baths. A fragment is placed in a sealed dish, then thawed for the first time in 30,000 years. What happens next is almost silent. Clear liquid is pipetted onto a plate seeded with amoeba, Acanthamoeba castellani, chosen for their vulnerability. Hours pass. At first, nothing. Then, under the microscope, the amoeba begin to round up and burst. The cells are dying, one by one, as if something inside the ancient ice has awakened. Genomic sequencing confirms the culprit, a giant DNA virus. Its genome scarred by age, C to T transitions, short fragments, all the hallmarks of deep time. But the virus is alive. It infects only amoebae, not mammals. The team repeats the host specificity trials, running controls, confirming the result. No human cells react. Still, every step is logged, every tube double-checked. The chain of custody is airtight. Biosafety level 2 is enough for now. But the question hangs in the filtered air. If one virus can survive this long, what else is waiting in the thaw? For the first time, the threat is not theoretical. It is growing, multiplying, right there in a dish. Proof that the ice is not just a vault, but a reservoir. And no one can say what may come next. At first glance, the fragments looked like any other bones pulled from the frozen ground. Bison, horse, the usual cast of Ice Age giants. But under a microscope, the story changed. Fine, parallel lines scored the surface. Marks so sharp, so regular, they seemed unlikely to come from the jaws of a carnivore or the slow crush of shifting ice. Some called them tool marks. Others were not so sure. Dating protocols left no room for error. Collagen was carefully extracted, purified, and run through accelerator mass spectrometry. The calibrated result was roughly 13,800 years before present. That is older than the Clovis horizon, by at least 6 centuries. If these marks were made by humans, it would mean people reached Alaska long before the textbooks allow. But in this field, skepticism runs deep. Could ice fracturing mimic the work of a blade? Could scavengers leave grooves that fool even experienced eyes? Microwear analysis became the battleground. Researchers compared the microstriations to experimental cuts made with stone tools. Some patterns matched, some did not. The debate turned forensic. Every groove measured, every edge cataloged. No one could agree. The evidence pressed at the edges of what science could prove. In the end, the bones raised a question that refused to die. Were humans here, hunting, butchering, leaving their mark in the ice long before anyone expected? Or was nature itself the artist, etching secrets into bone for us to misread? The answer, for now, remains locked in the details, waiting for the next fragment, the next test, the next challenge to what we think we know. At first, the ice core seemed lifeless, just a column of frozen soil, plants, and dust. But under the microscope, a different world flickered into view. Pollen grains, each one a fossilized fingerprint, told of vanished landscapes. Sagebrush, yarrow, then a sudden rise in grasses. The sequence mapped a slow motion collapse, cold steppe fading to tundra, forests shrinking, the old order breaking down. Extracted DNA added another layer. More than 800 distinct microbial signatures, bacteria, fungi, even ancient archaea, sat dormant in the permafrost, frozen in mid-activity. Some of these microbes have no living relatives. Others carry genes for survival in cold and dark. Their metabolism paused for 30,000 years. Each OTU, each taxonomic unit, is a name on a list of the lost. The story deepened with isotopes. In the collagen of mammoth bones, a subtle uptick in nitrogen, delta 15N, signaled a final, desperate winter. Forage had grown scarce. The plants changed, the soil dried, and the great animals ate what they could find. The numbers whispered of starvation, of a world closing in. Everything needed to reconstruct an ecosystem, pollen, DNA, isotopes, was locked in a single core. The ice preserved not just bodies, but the memory of collapse. And as the permafrost thaws, these archives come back to life, offering a glimpse into a past that feels too familiar. A warning written in code and bone, that the end of an age can happen quietly, one shift at a time. At first, the core sample was nothing more than a smear of dark grit pressed between glass. But under the Raman spectrometer, a sharp signal cut through the noise. A G-band at 1,580 centimeters to the -1. Materials scientists in the lab paused. This was the fingerprint of ordered carbon, but not graphite, not soot. Transmission electron microscopy went deeper and revealed particles just 2 to 5 nanometers wide. Perfectly round, dense, and scattered through the ice matrix. Nanodiamonds, or something close. too small to see with the naked eye, but unmistakable under the scope. Replication trials ruled out contamination. Lab blanks stayed clean. Still, no one could explain how such structures formed at these pressures and temperatures. The data did not match any known geological process. In the silence of the lab, the discovery felt like an intrusion. It was a message from a world where the rules had changed, and no one could say why. On paper, the numbers tell a simple story. Alaska's permafrost holds around 1,400 gigatons of carbon, twice what is in the world's forests, and nearly half of all soil carbon on the planet. Locked away, silent. But as the ground softens, that carbon is no longer safe. Microbes wake up. They chew through ancient peat, releasing methane, up to 1 gigaton trapped in hydrates, waiting for the ice to fail. Methane is a greenhouse gas with a punch. It heats the atmosphere more than 20 times faster than carbon dioxide over a century. Climate models try to predict what happens next. Most cannot keep up. Ice cores show temperature jumps of 2 degrees Celsius in a single decade, triggered by sudden releases. The feedback loop is brutal. More thaw, more gas, more heat. A climate modeler stares at the data, past and future stacked together, and admits the margin for error is wide. The real danger is not just in what is buried, but in how quickly it can change the world above. The Nagoya Protocol sets a clear rule. Any genetic material, ancient or not, requires benefit sharing and consent. In Alaska, every sample is bound by federal permits, biosafety reviews, and the expectation that indigenous voices are heard. Some scientists see ancient DNA as data, disconnected from living claims. Others argue that if profit or patents are possible, the descendants deserve a say. Indigenous leaders insist on consultation as a right, not a gesture. Biosafety protocols escalate. BSL-1 for plants, BSL-2 for viruses, and BSL-3 if mammalian infection is possible. Each step slows research, but never erases risk. Every expedition now answers to regulators and tribal councils. The next decision will shape the boundary between discovery and responsibility. Tonight, the ice keeps melting. Each year, new secrets surface, some beautiful, some dangerous, all still unexplained. Scientists warn that what is emerging could rewrite our history or threaten our future. The ground beneath us is shifting, and what rises next from Alaska's thaw may be stranger than anything we imagine.